Good morning, everyone. Welcome back to my channel, Molecular Mysteries. Hope everyone is doing fine out there. So, without any further delay, let's get into the class.
Come and fly away with me. Come and fly away with me.
Hello, everyone. Welcome back to my class. So, how are you all doing? So, hope you got a clear idea about glycolysis and the net gain that we produced after the glycolysis. Okay, so glycolysis is an important pathway which generates ATP and pyruvate, which would enter into the Krebs cycle for the generation of further conversion, and finally they would enter into electron transport for the ATP production. What is beta oxidation pathway? Before I want to talk about the Krebs cycle, I want to talk about the main important metabolism that happens in all the living cells, which is called a fatty acid metabolism. Okay, this fatty acid, what are fatty acids? Before you enter into this beta oxidation pathway, you should be aware of some important terms that we happen to hear frequently in this class, so that it would be clear for you to understand the concept so well, right? So you all know that, as you have already noticed, the thumbnail has given you that why exercise is a must for a person, like why physical workout is so important for a person. Because so you need energy, right? For whatever the work that you do, you need energy, and energy is stored in the form of fats. Also, you also derive energy from the stored fats, but you have to work it out, like you have to work out, then only these fatty acid metabolism would happen so rapidly that your triglyceride amount in your blood would be utilized for your ATP production. So before I enter into this, let me be clear about the terms called triglycerides. What is triglycerides? So you happen to hear this a lot in your blood test and all, right? So these triglycerides are important test for you how the fats are in your body. Like fats are stored in the form of in triglycerides. Okay, so you all know that adipose tissue is a tissue which contains fat cells in it, right? These fat cells mainly composed of glycerol with three molecules of fatty acids attached to it. Okay, so these fats have to be metabolized for the energy production. They have to be completely oxidized so that they can enter into the Krebs cycle for further mechanism to happen. Okay, so like glycolysis, it's like glucose breakdown. Fatty acids should also be broken down for the energy production. So this class is all about the breaking down of your fatty acid. Okay, so I told you triglycerides are something that they are the stored fat cells. Okay, they are mainly present in the adipose cells. These are all triglycerides are all present in the form of triacylglycerols, which are present in your adipose cells. So whenever they need energy, or whenever they require energy, and when they are under workout, this particular cells which are stored as fat or as triacylglycerols need to come out of that and get transported into bloodstream first. With the bloodstream, with the use of blood, they can get transported to the target cells where the ATP can be generated. Okay, so this beta oxidation pathway has got three important steps, where first of all the fatty acids have to be removed from like adipose tissue. They can first enter into the blood, and then to the cytoplasm of the cells, and then to the mitochondrial matrix where the beta oxidation would happen. Okay, let us go in detail about each and every cycle. Okay, so I told you adipose tissue, glycerol. So glycerol is a main triacyl glycerol is mainly composed of glycerol molecule, but added with three important fatty acids. Okay, and what is lipolysis? Yeah, this glycerol has got a very good affinity with the fatty acid chains, and it get regularized with that only. So lipolysis, with the help of an enzyme called lipase, helps in breaking down of that glycerol with three fatty acids, so that the fatty acids would be free form, can be easily entering into your bloodstream, right? So that is why lipolysis happens. So fat metabolism, I told you, that is a breaking down your fat for your energy production. And structure of fatty acid, why the structure of fatty acid need to be discussed? Because says, if you see the structure of fatty acid, you have got R group attached with the COOH group. Okay, right? And that R group depends upon the number of carbon atoms that are attached, the series of carbons that have been attached to that particular R group. Okay, and it could be 14 carbon compound, it could be 16 carbon compound, it could be 12 carbon compound, like that. So the carbon atom that is immediately located next to its functional group, is called the alpha position. At the next carbon next to alpha is beta position. So what happens in this beta oxidation pathway is there is a cleavage happening at the beta position of your carbon atom. So that part gets metabolized. They go enter into repeated cycles of your oxidation pathway, and this chain keeps on the keeps shorter and shorter. The number of carbon chains keep getting shorter and shorter, finally producing your acetyl CoA. Okay, I will tell you what are the key important steps in. That's why I want to talk about the structure of fatty acid here in the basic terms itself. Okay, so now I feel you got a little idea about the basic terms in beta oxidation pathway. Okay, let's go for the what are the steps. I told you the transport happens of fatty acids from the adipose tissue to the target cells, right? So the adipose tissue is full of fat cells which need to be transported through the blood into your target cells for your ATP production. So once they enter into the blood, they can immediately be diverted to your target cells and entering into the cytoplasm of the cells and finally into the matrix membrane, where the oxidative catabolism would happen. I told you the third important step is oxidative catabolism in mitochondrial matrix. So you know that mitochondrial matrix has got many enzymes because the Krebs cycle happens there, the beta oxidation pathway happens there, because all the enzymes are involved for the oxidation into your FADH and NADH2 so that they can enter into the immediate immediate area next to the matrix membrane is the inner membrane where the ETC happens, electron transport chain would happen. So after this class, I will start telling about you the Krebs cycle so that the both carbohydrate metabolism and the fatty acid metabolism would enter into the Krebs cycle. Then it leads to the so let it be clear, let it be slow. So let it be clear, let it well aware of all the enzymes and net gain in all the pathways. Okay, I will also link everything in detail in the description box. Please go through it. Okay. I told you triacylglycerols are hydrolyzed by hormone stimulated lipase. Lipase, I told you, they are the one which will break the glycerol molecule into free fatty acids for it to enter into the bloodstream, right? So for that to happen, there is a mechanism where the cyclic AMP gets activated. Okay, there are some hormones like glucagon and epinephrine which would trigger the receptors of your adenylate kinase. Okay, so this cyclic AMP will stimulate the protein kinase A and helps for the release of phosphorylation of your lipase. And thus the hormone sensitive lipase would come into the action, which would phosphorylate into triacylglycerols forming free fatty acid. So this slide is all about how the lipases would come into action, breaking the triacylglycerols into free fatty acids. Okay, now your free fatty acid, see this is your fat cell, triacylglycerol. So this is done by your lipase enzyme which breaks the glycerol and fatty acids, making it a free fatty acid. And this free fatty acid would definitely go for the beta oxidation pathway where it is produced as acetyl CoA. Okay, and the fatty acids getting converted into acetyl CoA and finally entering into the Krebs cycle where it's completely oxidized into carbon dioxide. Okay, and here you have glycerol in it. I told you glycerol would either enter into the glycolysis for the conversion into pyruvate, or it may either go for the gluconeogenesis. I told you gluconeogenesis is the production of glucose from a non-carbohydrate precursor, right? So this is called gluconeogenesis that happens in the liver cells. So for your brief information, this is what happens in the beta oxidation pathway and what happens here when the glycerol gets broken down from its fatty acids available for all the cycles to happen. So hope you got a brief idea. Okay, let's go for the second part of your cycle. What is that? Transport of fatty acids from adipose tissue to target cells. I told you once they are ready, the free fatty acids are now ready, and these free fatty acids should be released into your blood plasma. But you all know that the fats are not soluble in water, right? Like soluble in blood also. So for that to happen, a serum albumin that is also already present in the blood would act as a carrier for it. So it will bind to it, making it available for the movement of your fatty acid towards your target cells. So now it has entered into the target cells. Here comes the picture where it has to enter into the cell's cytoplasm. Okay, I will tell you what. So when the free fatty acid, before the free fatty acid should enter into the target cell, it has to be activated. This activation is the activation of that particular fatty acid is nothing but conversion into the acyl CoA. Okay, the acyl CoA is the only derivative, or you can say only form of your fatty acid where the mitochondrial matrix would accepts it for the oxidation to happen. Okay, every free fatty acid should be converted into acyl CoA. Then only it can be accepted by the mitochondrial matrix for further reaction. Okay, for that activation, there are some enzymes called acyl synthase. Acyl CoA synthase would help for the activation of your free fatty acid into acyl CoA. So now you are ready with your acyl CoA. This acyl CoA can immediately enter into mitochondrial matrix, but it has got a very complex structure. I will let you know in the next slide what really happens, okay, and what's the process, how it enters into it, and what are the enzymes that are involved here. Okay, so here comes the importance of carnitine shuttle. What happens is, I will tell you a brief diagram of it. So before I enter into the beta oxidation main pathway, there is a clear complex where the mitochondrial matrix would accept that acyl CoA only in the form of carnitine acyl CoA. So what is that and what is carnitine shuttle? So here I have given you a small brief idea picture that I want to talk about. Okay, see, this is your outer mitochondrial membrane and this is your inner mitochondrial membrane. Okay, now, and this is your cytoplasm as usual, and this is your mitochondrial matrix is present here. Now what happens, you have your acyl CoA ready. It is already in the cytoplasm. Now it has to enter into the matrix for your beta oxidation to happen. For that, acyl CoA is easily permeable for your outer membranal matrix. Like outer membrane of your mitochondria is permeable because it has got porins in it. Porins would easily help for the acyl CoA to enter into it. But this inner membrane is not permeable for this acyl CoA. This inner membrane does not allow the acyl CoA to get entered into matrix. So for that to happen, you got a complex called carnitine palmitoyl transferase 1. So what is that? It's a carnitine shuttle. I can say you in short, it's a carnitine shuttle. These are the group of enzymes that are helpful for the conversion or for the movement of your acyl CoA into your inner mitochondrial matrix. Okay, this carnitine palmitoyl transferase 1 is the name of an enzyme that is present in the outer mitochondrial membrane. Okay, and this carnitine acyl carnitine translocase is another enzyme present in the inner mitochondrial membrane. Okay, so once the acyl CoA has entered into the, this is called IMS, intermembranal space. Okay, so once the acyl CoA has entered into it, it cannot enter to the inner membranal mitochondria. So here what happens is, this carnitine palmoyl transferase would help to release of carnitine, and thus the carnitine binds with the acyl CoA, forming carnitine acyl CoA in the intermembranal space. Once it is formed, it is released out or released inside. It is released into your mitochondrial matrix by as acyl carnitine. Okay, you got the acyl CoA is converted into acyl carnitine in your mitochondrial matrix, and this CoA is again released into the cytoplasm for the further usage. Okay, now this acyl carnitine that is present in the inner mitochondrial membrane, now you know that this carnitine has to be again regenerated and the acyl CoA has to be again regenerated for the further use. So here comes the enzyme called carnitine acyl carnitine transferase. This is the enzyme that plays an important role where the acyl carnitine is converted into the carnitine. Okay, this carnitine is again utilized by the acyl CoA so that it again converts itself into acyl carnitine, allowing the acyl CoA to enter into your matrix. Okay, so what is carnitine shuttle? This particular enzyme, it is helping the acyl CoA to enter into the matrix through carnitine. Okay, once this acyl carnitine is converted into carnitine, this carnitine is again transported back into the intermembranal space, and this intermembrane space where the acyl CoA has already entered binds with that, forming acyl carnitine, and enters again to the matrix. And this acyl CoA is again regenerated back into the mitochondrial matrix. So you got your acyl CoA, which where the mitochondrial matrix will undergo for the beta oxidation. So you got your acyl CoA, and this you got your carnitine getting cleaved, and this carnitine again goes into the intermembrane space, and in where it attracts acyl CoA, forming acyl carnitine. So this is the cycle that happens here. Okay, the whole cycle happens with this conversion. Hope you are clear with the diagram. So I will also share you in the description box below. Okay, for your understanding I have drawn a brief diagram. If you want to have better understanding, you can go through the slides, and important structures are also attached in your description link. Hope you're clear with this of your mitochondria. So now it has prettily entered into your mitochondrial matrix. So now it's a time for us to talk about what's happening in the mitochondrial matrix. Okay, now so you are pretty well clear about it. Okay, next go for third step. The third step being oxidative catabolism in mitochondrial matrix. So now you got your acyl CoA. Acyl CoA is already there for your oxidative catabolism to happen in your mitochondrial matrix. So I told you acyl CoA, the structure of acyl CoA has got the H part of your acyl CoA is being replaced with S CoA, and also the fatty acid CoA depends upon the number of carbon atoms atom. I told you the fatty acyl CoA, the R group, depends upon the number of carbon atoms that are present. It could be 14, it could be 16, it could be 12, like that. So if I am taking for example in this class I want to talk about the palmitoyl CoA, which is a 16 carbon fatty acid chain. Okay, this 16 carbon fatty acid chain gets go for repetitive oxidative cycles, making the chain shorter and shorter. Thus it will end with the cycle producing the acetyl CoA, which goes for the further Krebs cycle. Okay, the main important end product of your beta oxidation pathway is the acetyl CoA production. Okay, the conversion of your acyl fatty acid into acetyl CoA. Once the acetyl CoA is produced along with FADH, NADH, they enter into the Krebs cycle, where the acetyl CoA gets converted into completely oxidated to CO2, producing NADH for the further use. Okay, so every cycle of, so I told you the beta oxidation pathway happens, it keeps happening on the fatty acid chain that is added to the acetyl CoA. Okay, this fatty acid chain, upon number of repetitive oxidative cycles, it the chain gets shorter and shorter, so it goes again to the main cycle, and again goes down, and again produced, so that maximum number of carbons are being utilized. Okay, for the production of acetyl CoA. By the end of fatty acid metabolism, what for the 16 carbon compound like palmitoyl CoA, it produces 8 molecules of your acetyl CoA. Okay, so how the pathway happens, this is the simple pathway that I want to talk to you. So here I have taken the palmitoyl CoA. I told you this is the alpha position, which is immediately attached to your functional group. Okay, and this is your beta position. I told you this is how, where the cleavage happens, that is called as beta oxidation pathway. This particular chain is a 16 carbon compound, of course it is a palmitoyl CoA I'm talking about. And now the acyl CoA dehydrogenase would convert itself into enonyl CoA, where there is a release of FADH2, one molecule of FADH2. In the same way, this enonyl CoA gets converted into hydroxyacyl CoA with the enzyme called hydratase, with the addition of H2O molecule. Okay, and the same way, this beta hydroxyacyl CoA would again go for another dehydrogenase enzyme reduction, forming beta ketoacyl CoA, releasing of NADH. So for each and every cycle you have a release of one molecule. For a one cycle of your beta oxidation, there is a release of one FADH2, one NADH, right? So let us now calculate the net gain also. Okay, see here, once conversion of it enters into the beta ketoacyl group, and then it transfers into thiolase. With an enzyme called thiolase, you get acetyl CoA and ministryl CoA. Yes, see now here it started with a 16 carbon compound called palmitoyl CoA and ended with a 14 carbon compound. That means the two carbons that have been in palmitoyl CoA is now shifted to acetyl CoA. Right, here are the two carbons. So what happens if you start with 4, like for example, if you started this beta oxidation with a ministerial CoA, that is C4, nothing but 14 carbon compound. Out of the 14 carbon compound, after one round of your beta oxidation, you will get a 12 carbon compound with a 2 carbon acetyl CoA. In the same way, if you start with 18 carbon, it produces 16 and 2 carbon compound. If you start with 16, it gives 14 carbon compound of menstrual CoA and 2 carbon compound acetyl CoA. That is how. And this menstrual CoA again goes for your oxidation, where it starts with 14 and ends with 12. After getting that, again the 12 carbon compound would enter into the oxidation pathway and again produces 10 carbon compound. That is how the breaking down of your fatty acid molecules would happen till it produces the number of acetyl CoA. So this is the term that I want to see. This C14 would become C12, and C12 would again become C10. I mean, it's a 10 carbon compound, 8 carbon compound, 6 carbon compound. In that way, you will be producing lots and lots of acetyl CoA, which is the final product of your beta oxidation pathway. This acetyl CoA would now enter into your Krebs cycle. So, in the next class, I will talk about what happens in the Krebs cycle. Okay, so this is acetyl CoA which enters into Krebs cycle, and this so let me talk about the net reaction. For a carbon compound like 16 carbon compound like a palmitoyl CoA, for complete metabolism of your 16 carbon compound, it has to undergo the cycle 7 times. The 16 carbon palmitoyl CoA, for production of 8 molecules of acetyl CoA, has to run 7 times. Okay, so for imagine, after 7 cycles of your beta oxidation, we get 8 molecules of acetyl CoA. One molecule, like for one molecule, for one cycle you'll be getting one molecule of ATP. Just now we have seen that only one ATP is, one NADH is produced and one FADH are produced for one cycle. So if this cycle is happening 7 times, like 7, it's running 7 times, obviously it gives 7 NADH, 7 FADH with a net gain of 8 acetyl molecules for one molecule of carbon 16 carbon palmitoyl CoA. Hope you're clear with it. If you have any confusion, please let me know in the comment. Okay, I will let you know the in detail structure, and also I will also provide you the importance of the enzymes in this pathway also. Okay, so hope you are clear with the beta oxidation pathway, how the fatty acids get metabolized. Okay, so see you in the next class. Until then, stay positive, stay healthy, and be kind to everyone. Thank you. Bye bye.